Welcome students. Today we are discuss about nutrients of biofertilizer. Means it is another formulation of the biofertilizer. Earlier we are discuss about the carrier based biofertilizer, but today we are discuss about the liquid based biofertilizer. So there is two type of biofertilizer. First one is carrier based and second one is liquid based. So what is the definition of liquid based? What is the need of the liquid based biofertilizer? And what is difference between carrier based and liquid based? How it is advantageable for the farmers? कैसे लाभदायक है ये? So today we are discuss all these things. What is the shelf life? What is the how to protect the how to restrict the multiplication period, multiplication time. So let us start. The nutrients of biofertilizer. Under this banner we are discuss about the liquid biofertilizer. And what is the liquid biofertilizer? It is a liquid formulation of the containing not only desired microorganism, but it is a some special cell protectants which is give the resting period of the spores. It means you can say the cell protectants and other chemicals are very useful to give the microbial cells in the resting period with the effect of their chemical properties. So it is a desired combination of the desired microorganism and their nutrients, but also a special cell protectant which is help to resting the spores are substances that encourage formation of the resting spores or make a cyst. That is a cyst, you can say it is a dormancy period of the cells, microbial cells, for longer shelf life and tolerance to the adverse condition. As you know, in the rhizosphere there is a very too much factors is affecting the microbial population of the native rhizobia and other rhizobium, Astobacter, Azospirillum, etc. So what is this nutrients and how to formulate this liquid biofertilizer using this unconventional synthetic materials like artificial food for that is useful for the microbial cells. So these are the polymers have demonstrated potential as bacterial carriers that afforded substantial advantages. Like you can say formulation encapsulate in the form of capsule with the help of the some fillers of the like living cells, protectant and microorganism against many environmental stress and release them to the soil gradually but in a large quantities. Second one is when the polymers are degraded by the soil microorganism, usually at a time of the seed germination and seedling emergence. Third one is they can be stored dried at ambient temperature for prolonged periods after a consistent batch quality and the better defined environmental for a bacteria and can be manipulated easily according to the needs of the specific bacterial cell. These inoculants can be amended with the nutrients to improve the short-term survival of the bacterial upon the inoculants and it's give the longer life of the bacterial cells present in this liquid formulation. So how to formulate this liquid biofertilizer in the form of the encapsulation in capsule. So the capsulation of the microorganism into a polymer matrix is still experimental. At present it is a experimental basis and it is research on this topic is going on, how it is make very beneficial and increase the shelf life of the microbial inoculants. So it is under process. At present there is no commercial bacterial product using this technology because it is not perfect the technology in the India. So it is under progress, and I think 2 or 3 universities are manufacturing this type of liquid biofertilizer, but largely maximum universities, agriculture universities, are using carrier based or manufacturing carrier based microbial inoculants. Then third one is the concept underlying immobilized microbial cells is to be entrap beneficial microorganisms into the matrix. As you know, in the rhizosphere there are so many bacterial, fungal and actinomycete like microorganisms available. They are competing with this nitrogen fixer, phosphate solubilizer and micronutrient mobilizer. So there is a competition in the rhizosphere in between artificial inoculation and native rhizobia. Then formulation of the bacterial matrix is then fermented in a bacterial growth medium under the some certain specific chemicals which is provide the shelf life of the cells and formation of the cyst in the liquid as a dormancy period of the cells. That's why it's give the long life, more than 1 year for this microbial culture. This formulation can produce many useful compounds for industrial and environmental application such as organic acid, amino acid and enzymes. The biodegraded toxic material, you can say bioremediation over extended period of the time. So all these are able to produce a cyst form of the microbial inoculants in the liquid culture to increase their efficiency and increase the shelf life. So first one is alginate. You can say it is a type of filler. Alginate is a material most commonly used for encapsulation of microorganisms. It may be nitrogen fixer, it may be phosphate solubilizer, it may be micronutrient mobilizer, etc. These resulting inoculation are used for various purposes like immobilization of the cell organelles and enzymes, the application of bacterial control agent and mycoherbicides, and third one to increase the stability of the combination of plasmid in the host cell. So with the help of alginate you can make a form of bacterial cell in the form of capsule for the use of agricultural use. Then what is the role of this liquid biofertilizer and how it is differ from the carrier based microbial inoculants? So the role of this liquid biofertilizer is they supplement chemical fertilizer for meeting the integrated nutrients demand of the crop, especially chemical fertilizer. Application of biofertilizer result in increase the water uptake, root development, vegetative growth and nitrogen fixation from the atmospheric nitrogen. As you know, in the atmosphere about 79 to 80% nitrogen is useless for the plant. So through this nitrogen fixing bacteria you can conserve in the soil. Second one is application of biofertilizer results in increase not only the plant nutrient as well as its help to check the some fungal effect to the in between crop cycle periods. They act as an antagonist and suppress the incidence of the soil-borne diseases, pathogen, and thus help in the biocontrol of the disease. So nitrogen liquid biofertilizer is not only to help the save the nitrogenous fertilizer, but also help to check the soil-borne diseases. They can add about 20 kg to 200 kg of the nitrogen in case of nitrogen fixers under the optimum cell condition, and thereby increase crop yield by 10 to 15% of the total crop yield. Next role is the liquid biofertilizer is some biofertilizer like rhizobium, blue-green algae, Azotobacter, phosphate solubilizing microbacteria stimulant the production of the growth of promoting substances like vitamin B, vitamins like vitamin B complex, indole acid and gibberellic acid. These hormones, these vitamins are helpful for the plant growth and increase the crop yield as you expect from the recommended dose of the fertilizers. Next role is the phosphate solubilizing bacteria, or you can say phosphate solubilizing microorganism. It is a two type. It may be bacteria, it may be fungi. So this phosphate solubilizing or mobilizing fertilizer convert the insoluble soil phosphate. Insoluble soil phosphate means in the form of tricalcium phosphate. It is not very less soluble and not available to the plants. So it's converted into dicalcium phosphate or in the soluble form that secretes several organic acid, and under optimum condition they may solubilize or mobilize about 30 to 50 kg of P2O5 per hectare, due to which crop yield increase by the 10 to 15%. So mainly the insoluble form converted into soluble form, then after plant is absorbed as a nutrient. Next one, the liberated growth promoting substances. It is a very important character of the phosphate solubilizing bacteria. It secretes some plant growth promoting substances like vitamins and help to maintain the soil fertility as per the need of the crops. They are cheaper source of the nutrient. It is a pollution free, and it is, you can say, it is a renewable energy of the source. They improve the soil fertility and soil productivity and play an important role in the cycling of the plant nutrient. So altogether it is helpful for the soil health as well as increase the crop productivity. Next slide is showing what is the benefit of the liquid biofertilizer. What happened? How to save the chemical fertilizer after using this liquid biofertilizer. So first one is with the help of liquid biofertilizer over the conventional carrier material like lignite based, charcoal based, compost based biofertilizer. The shelf life of the liquid biofertilizer is more than 12 months, means 1 year. Whereas in the carrier based biofertilizer, the shelf life of the microbial cell is only maximum 6 months. So farmers can't use next year because the carrier based microbial inoculants is not survive after 6 months in the carrier available carriers, but in the liquid based biofertilizer it gives more than 1 year, 1.5 year their shelf life, and farmers can use in the next year. If not able to use current year, they can use in the next year. There is no contamination was recorded in the liquid biofertilizer, so it is a contamination free fertilizer. It is easy to identify by the typical fermented smell. liquid biofertilizer is having a very good separate smell rather than other carrier based microbial inoculants. It is also better survival on the seed and soil. So if you use either seed inoculation or in the soil application, so survival rate of the liquid biofertilizer is high comparison than the carrier based microbial inoculants, because in the rhizosphere so many factors including native rhizobia also effect also give a adverse impact on the liquid biofertilizer, and that situation it is fight against this native rhizobia and work for the crops. It is a very easy to apply comparison than carrier based biofertilizer. We will discuss in the next slide how to apply in the field. Then it is the high commercial revenue. If you start internship of the commercial level biofertilizer production, so liquid biofertilizer is better, and I think give a better revenue comparison than carrier based biofertilizer. So you can also adopt as your startup program or entrepreneurship if you interested. Then it is traditionally liquid biofertilizer produced from the fermentation of effective microorganisms was recommended to be used within a year or so, for 14 to 16 months. Liquid biofertilizer known to influence nodulation, symbiotic nitrogen fixation, and growth and yield of the pulses if you are talking about the nitrogen waste, nitrogen fixing microorganism. They reduce the use of chemical fertilizer by maximum 15 to 40%. So assume if you save the chemical fertilizer about 40%. So nowadays chemical fertilizer is very costly. DAP is about 12 to 15 rupees per kg. Urea is 6 to 7 rupees per kg. So you can save the chemical fertilizer and also save the money after involvement of this liquid biofertilizer. They have long shelf life and easy to produce and apply. So it is slightly easy to application in the soil, like the carrier based fertilizer, carrier based microbial inoculants. Carrier based microbial inoculants need to prepare as agri solution and then cool down, then mix and apply in the field. But liquid based biofertilizer is very easy to apply in the field. Then it is tolerant to the temperature and contamination free, whereas in the liquid carrier based biofertilizer it is a temperature sensitive. Its temperature goes up to 45. It is a very tough for the microbes. After 45 degrees centigrade, the population of the carrier based microbial inoculants is reduced from 10 to the power 9 to 10 to the power 6. So in this condition, liquid biofertilizer is better than this carrier based biofertilizer. Then how to apply the liquid biofertilizer? What is the methodology? How to use the seed inoculation, root deeping and soil application, and which type of ways is available for this liquid biofertilizer? This is a 3 ways for the liquid biofertilizer. First one is seed treatment, then root deeping, and then soil application. That is similar with the carrier based biofertilizer. So come to the seed treatment. How to apply the liquid biofertilizer with the seed in the field. The seed treatment is most common method adopted for all type of inoculants. The seed treatment is effective and economic. For a small quantity of the seed, up to 5 kg quantity, the coating can be done in the plastic bag. For this purpose, a plastic bag having size 21 into 10 inch or big size can be used. The bag should be filled up with the 2 kg or more seed as per the capacity of the bag. The bag should be closed in such way to trap the air as much as possible. The bag should be squeezed for 2 minutes or more until all the seed are uniformly coated like a black color. The bag is opened, inflated against the sect gently, stop shaking after each seed getting a uniform black color layering. The bag is opened and seed is dried under the shade for 20 to 30 minutes. Then apply in the field. For the large amount of the seed coating can be done in the bucket, and inoculant can be mixed directly with the hands. Seed treatment with Rhizobium, Azotobacter, Azospirillum along with the phosphate solubilizing bacteria can be done with the same method. Then root dipping. For the application of Azospirillum, Azotobacter, phosphate solubilizing bacteria in the upland paddy, not transplanting paddy, vegetable crop may be used. This method is used through this root dipping. The required quantity of the Azospirillum or any other microbial inoculants has to be mixed with the 5 to 10 liters of water at one corner into the field, and the root with seedling has to be dipped for a minimum of the half an hour before transplanting or sowing. Then soil application. Soil application, under the soil application 200 ml of microbial inoculants mix with the 400 to 600 kg of cow dung or FYM along with the half bag of the rock phosphate if available, because it is a very good source, very cheap source of the phosphorus. As you know, phosphorus is a major component of the plant nutrient. The mixture of the PSM with the cow dung and rock phosphate had to be kept under any tree, or you can say shed, or under any set condition over the night and maintain 50% moisture. Why over the night? Because the cell is under the dormant period in the liquid biofertilizer. If we apply with the compost, FYM and others, this dormant cell is start activity very fast, and it will take about overnight, means 12 to 15 hours. Then after activation of the cyst cell, you can apply in the field. The mixture of the PSM, cow dung, rock phosphate had to be kept. It is very important with the maintaining of the 50% moisture. Use the mixture as the soil application in row or furrow, or during leveling of the soil, it means before the plowing, or at the time of plowing you can also open the furrow and apply this biofertilizer mixed with the cow dung. Then what will the doses of the liquid biofertilizer as per the pulses, cereals, etc. So in case of pulses like green gram, chickpea, black gram, pigeon pea, etc., if you are going to use Rhizobium, PSB or Azotobacter, the application method 5 ml per kg of seed. Remember this point, 5 ml per kg of seed is sufficient for the inoculation if you are going to do seed treatment. Then in case of cereals like wheat, oats, rice, etc., the Azotobacter and Azospirillum is very important. The amount is the 5 ml per kg, that is uniform for the pulses, oilseed and millets. This table shows the response of the liquid biofertilizer on the different crops, especially we are talking here about cowpea. So if you apply the Rhizobium culture and compare with the control, means no inoculation, the control yield is 10 quintal per hectare, whereas Rhizobium or PSB inoculated field give a 13 or 13.5 quintal per hectare. So increment is about 30-35%, whereas we are claiming 10 to 14%. On an average, it's give about 20 to 35% in the farmer's field. So these are the response of the biofertilizer in the cowpea. So question arises, which type of factor and other parameters affect the performance of the biofertilizer? First one is the host plant. Rhizobium culture is a host specific. The rhizobium culture, if rhizobium culture is for the pigeon pea, it is suitable and act only in the pigeon pea, not in the green gram. So it is a host specific, remember this. It is not for all. So pigeon pea, rhizobium culture not work in the cowpea and the green gram and others. So it is a specific, host specific. Then soil fertility, if soil fertility is very high in your field, like nitrogen, phosphorus and potassium is very high, so the performance or effectiveness of the any microbial inoculants is suppressed by this fertility. Then cropping practices. Cropping system is a very important. So crop rotation is very useful for this microbial inoculants. If you are using cereals, then pulses, it is good for this inoculants, microbial inoculants. Neither if you are using always cereal which is a nutrient exhaustive crops, at that time like rhizobium or a PSB is performing very good, because the nutrient available in the soil, into the soil, is absorbed by the plants. So deficiency was created in the soil. In that condition, this any microbial inoculants is working very good. Then microbial and environmental factors, biological and environmental factors. So if temperature is high, rainy water always in submerged condition, so performance of the biofertilizer, especially nitrogen, PSB and Azotobacter is not working very good, because it is a aerobic bacteria. So if you provide the submerged condition, it is not work very good. So survival and efficacy of the biofertilizer are affected by the above factors. We are recommending some important books for you in the Indian condition, Indian books. Biofertilizer in Agriculture and Forestry, it is written by the Dr. N.S. Subbarao. It is very good for this my agricultural microbiology. Then Biochemistry of the Biotechnology of the Biofertilizer, written by Dr. S. Kenyon, ex-Vice Chancellor, Tamil Nadu Agriculture University. So you can read this book. Another book is Recent Advances in Biofertilizer Technology by the A.K. Yadav and others. And next book is Phosphatic Biofertilizer. This book is deal with the only phosphatic microbial inoculants, phosphatic bacteria, fungi, etc. So you can refer this. So these are the important book for this course. Thank you.